First, I have to give the greetings of our Vice Rector of Postgraduate and Research, Hernan Maureira. He sent in the greetings and give all the well best to this presentation of ours, particularly the end of the very interesting project that we have in FOVI. And due to they asked me to give this presentation in English, sorry for the non-speaker, but we have a very well renowned researcher invited today. This is our university is trying to develop more international collaboration in different areas of the science. This project is part of a neat project that from like 2 years now is becoming better collaboration, international collaboration between universities, and pay principally to invite researchers from abroad and also send a student and researcher to other universities abroad. Aparna is giving the end of a very interesting project that working in biopolymers. But also the first and most interesting thing of this kind of project is that we develop a network of collaboration internationally to bring very good researchers to our university here, the University Catholic of Maule, Universidad Católica del Maule, name that we not translate name of universities normally, and bringing researchers to here and put to our students good research and also provide the opportunity to all of our researchers, all of our academics can apply to this kind of project and send a student abroad. I have to say thank you to Aparna, thank you to Travana to provide us to this kind of meetings. Aparna now is also working with 2 years, now is working in Autónoma. She was part of our center CM, and now is bringing collaboration of 2 universities. So I have to say thank you very much to all the people here. Most welcome to international researchers that are coming and giving the talk today. And in the name of university, thank you very much, Aparna, and I hope it will be a very nice presentation. Thank you very much.
He will be speaking today on some of his work related to the polysaccharides, plant polysaccharide that he is working, and also its anti-cancerous activity that they are focusing. So we'll be starting with him.
Hello, everyone. So, as said before, I will give my speech in English because I'm pretty sure if I talk in Portuguese, you are not going to understand, as I can't understand Spanish also. So we are brothers in South America, but well, I speak Portuguese, but today I'm going to speak in English, okay? So today I'm going to speak a little bit about this topic, that is unveiling polysaccharides as anti-cancer agents and as a functional nanoparticle development enablers. It's very important for me to be here in Chile and to be collaborated with Aparna because she visited my lab and we got some really good results about what she's working with this kind of climate change thing. But I am working with more a food science approach. Okay, so what I'm going to speak today, it is related with some biological effects that polysaccharides that we extract and isolate from different samples, especially food, we can give to human beings for our health. So if you would like to scan this QR code, you can get my contact, but at the end, I will have another QR code that you can scan. So I am associate professor at the Department of Food Science and Experimental Nutrition at the Faculty of Pharmaceutical Science in University of São Paulo. So what I'm going to talk a little bit today is how we can imagine some biological effects through food ingestion and the polysaccharides that we have in some kind of fruits, and also the technological effects that we could have from these polysaccharides through the isolation of these polysaccharides, enhancing some kind of food supplements, and also making some nanocapsules. And I would like just to put this artificial intelligent image that summarize some of my researches, some of data that I will present today. It's very important for us to ingest fruits and vegetables. And why that? Because fruit and vegetables that they are regularly consumed could lower the risk of cancer death. This is because the presence of some kinds of vitamins, minerals, secondary metabolites, and the dietary fibers. They are all called bioactive compounds because they are good for our health. So what I'm going to talk a little bit is about dietary fiber. That they are mainly formed by polysaccharides, and they are divided in soluble and insoluble dietary fibers. But what I'm looking for is the soluble in water, of course, dietary fibers, because they can have some really nice health effects in our body. They are formed mainly by oligosaccharides, some kinds of glycans, gums, and the main bunch of molecules that I work in my lab, it's pectins. They increase the stool viscosity, they can reduce the glycemic response and plasma cholesterol, and they can be fermentable by human intestinal microbiota, producing short-chain fat acids that has already been seen a lot of biological effects in our health. But the thing is that polysaccharides as food fibers, they are really complex because they are not just a small molecule that are isolated, that is isolated. It's a mixture. It's a mixture of several kinds of molecules. So the thing is that what I'm studying in my lab is the correlation between the high molecular and the low molecular weight for increasing the biological effects, and also the high molecular and low molecular weight polysaccharides that could give some better technological effects. So why this is important? Because as we get the fibers, the polysaccharides, more soluble, we can increase their fermentability and we can increase the immunomodulation. So the high molecular weight polysaccharides, they are not 100% soluble in water. So they are not going to interact with some kind of receptors, and also they can't be completely fermented by our microbiota, by our intestinal microbiota. So we need to develop some kind of techniques to decrease the high molecular weight polysaccharides present in foods. So what we do, we modified our pectin. And I just brought it here, the modified citrus pectin, because it's a dietary supplement that it's sold in the US market. And this is, what is the modification of the citrus pectin? They only decrease their complexity. They decrease the molecular weight. So they increase the homogeneity of the samples. And this is very important because lots of researchers, including me, we could trace their anti-cancer effects through one main pathway, that it's the galectin-3 inhibition, that I will show that some of these pectins, the low molecular weight pectins, can show an anti-cancer effect through the galectin-3 inhibition. So the main focus of my work was how we could decrease the molecular weight of pectins from different kinds of food sources. So this is just an example of how pectin is complex. So in the same molecule, we have several types of glycosidic bonds, different types of sugars. It's a highly complex molecule. But the thing is that if they are high, if their molecular weight is really high, we can't extract the best biological effect. So what we need to do, just like the modified citrus pectin, so we cut it. We cut it in several ways to decrease their size, but the techniques that we use to cut it, it can be high temperature, using some chemicals, using some enzymes, or even leave the fruit to ripe. And what I'm going to show today, it's a model that we work in Brazil, that is a fruit that is called papaya fruit, that during the ripening, it expresses a lot of enzymes that will depolymerize the pectin for us. And the main question was, is the natural fruit ripening having the same biological effect than the modified citrus pectin? So the nature will change. The nature will modify this pectin for me. And why I did this question? It's because in my PhD I have studied the ripening process of the papaya fruit, especially looking and seeking for the plant physiology thing. So we identify several types of enzymes that cleave the pectins, and one of them was a polygalacturonase that we express in papaya fruits. We also saw that this expression is highly coordinated during ripening. We also saw that this ripening process is coordinated by several transcription factors, especially a NAC1, and this is regulated by some kind of epigenetics. And through a system biology approach, we could identify the main enzymes that will hydrolyze this pectin for us. And when I became a professor, the question was, can I increment the biological effect just leaving the papaya fruit to ripe? So this is just a model of the fruit ripening. Papaya has almost the same profile of a climateric fruit, just like tomato, and a different one from strawberry. So we were able to identify the main genes and protein that were found to be responsible for pectin solubilization. And what we did, the question, is the papaya ripening will increase the soluble fiber? And this is a very important thing, because what we saw in our experiment was that the pectin that was insoluble in water in ripe papaya, as it gets ripened, the pectin will get more soluble. So what we want soluble pectins. And then we started extracting and isolating the pectins from several ripening times in papaya, and we saw that the pectins extracted from the 3rd to the 4th and 50th ripening day, we showed in vitro anti-cancer activity through galectin-3 inhibition, that it was completely dependent on ripening and completely dependent on the pectin structure. So the main question was partially answered by our in vitro tests. We continue to isolate this kind of polysaccharide, so we passed the whole pectin through several types of chemical and physic isolation, and we saw that one part of the pectin that was isolated from the 3rd harvest time, the 3rd ripening time, has a very good in vitro anti-cancer activity through the inhibition of galectin-3, and it was just like the results were just like the modified citrus pectin. And we continue to isolate it through some, and we identified that some acid pectins that has some low molecular weight, they also have the in vitro anti-cancer activity through galectin-3 inhibition, and now we have proved it through the knockout of galectin-3 expression in our cells, and then we saw that the cells, they didn't die after the pectin treatment. So we got a very good correlation through the expression of galectin-3 and our pectins. And we had done just with cells in a bidimensional way, so we started creating our own three-dimensional structures of cells that were from colon cancer cells, and we put together some kind of fibroblasts to express protein from extracellular matrix, and we could mimetize what it happens in the microenvironment of a tumor. So we treated these small tumors that is called spheroids, and we treat it with our pectins, and we saw that the unripe, but mostly the ripe papaya, has decreased the growth of these spheroids. So we validated in a more complex method, in a more complex experiment. We continue to do that, and now we have a panel of spheroids that we can treat our spheroids, and we can see how many cells are dead and how many cells remained live after our treatment. And of course, we have all the controls. But we need to go further. So we start working with rats, and we induced the colon cancer, at least the pre-lesions of colon cancer in these rats, using azoxymethan, that is a carcinogen. And we separate in 4 groups: the non-treated one that we inject only saline, the one that was treated only with regular show that is it's made of microcrystalline cellulose, and then we had other 2 groups that were supplemented with green papaya fibers that contain pectins and ripe papaya fibers that contain more soluble pectins. And what we saw is that the number of lesions in the rats that were fed with our ripe pectins decreased. It's not that they were not having pre-neoplasics lesions. They have it, but it was in low number. And of course, we are not talking about a drug. We are talking about a food component that is completely different. So we don't want to mitigate the whole cancer process, but we can decrease the incidence of cancer by creating maybe a dietary supplement. And the good thing is that in the ripe group, we have an increase in the apoptosis level in these cells, mostly in distal part of the colon. And why it happens? We know that it could be something related with inflammation. So if we decrease the inflammation, we could also decrease the incidence of colon cancer. And we start seeking for the combination, or not the combination, the integration, the ligation of some parts of pectins with some parts of receptors that are intrinsically related with inflammation. That is called toll-like receptors. And we saw that the pectins extracted from different types of ripening times, they could modulate in vitro some toll-like receptors in a model of cells that express each one of these toll-like receptors. And because of this, what we did, we started seeking if our pectin could have a direct interaction with toll-like receptor, and this we saw just for the toll-like receptor 1 and 2, but we are running new experiments for the other toll-like receptors. And the breakthrough experiments, in vitro experiments that we did, we created some organoids from cells that were isolated from the terminal ileum from neonates, and they were cultured as an organoid. We break it, we put it in a bidimensional way, so we had a very specialized colon and intestine from neonates. And what we did, we challenged it with a E. coli that caused necrotizing enterocolitis in neonates. And we put together our pectins to see if we could decrease the inflammation driven by this pathogenic bacteria. And what we saw is that the pectins, they were not cytotoxic for the organoids, and they decreased the expression, especially of the interleukin 6. So this is quite interesting. We did it with a collaborator in University of Maryland, and now we are creating this kind of dietary supplements directly for neonates. And she's asking for permission to do it as a clinical trial, because in the United States they have this kind of, it's really high. We know why, but they have lots of neonates that they have this necrotizing enterocolitis. And why this happened? Because when they are born, they might not have their intestinal microbiota formed. Of course, we know that. But something happened that this bacteria will start an inflammation in their colon and their intestine, especially their intestine. And this is a cycle that gets really, it's a really fast process that necrotize their intestine. So they have to open and they have to take out their intestine. And this is mainly because of the presence of this bacteria. So if we gave this pectin to them, of course, it needs to be sterilized. All this procedure need to be followed. So if we do it, we could maybe take this neonate from this inflammatory cycle, because just giving antibiotics, it's not a good way, because it's really toxic, the antibiotics. So I showed you that the papaya ripening could increase the biological effects and maybe some indirect biological effects. I didn't show the modulation of microbiota, the intestinal microbiota, but just this kind of immunomodulatory I have showed you. But the thing is that the ripening process is not something that we control. Who controls is the fruit. So the best way now that we are doing in our laboratory is producing our own modified pectins. And we started doing by extracting and isolating from green and ripe papaya, green chayote that I know that you don't have here in Chile, but it's something that it's really common in Brazil, and it's full of pectin in this fruit. And we are also working with passion fruit, especially the albedo part. It's the mesocarp, it's the white part. Why is that? Because in Brazil we have a very big juice industry of passion fruit that they only use the pulp and everything is discarded. And the mesocarp is fully of pectin, but it's fully of high molecular weight pectin. So what we try to do is to modify these pectins to see if we had the same results of the modified citrus pectin. And why it happens? Imagine that we have this is maybe a pectin that it's not soluble in water, and just if you try to extract with water, you are not going to extract lots of molecules. But if we did some kind of chemical or enzymatic treatment here, we can break the glycosidic bonds and we can enhance the number of molecules extracted in water. It's just like this. It's a schematic here. So we have some pectins that are soluble in water, but they are connected with insoluble part. So when we treat it with chemicals, thermically treatment, and enzymatic, we can cleave this and we can release the soluble pectins. We try a new thermally modification using the citrus pectin, and it was a breakthrough method that I will show you that we applied for other types of fruits, and our own modified pectins by this breakthrough treatment had shown an anti-cancer effect, no contamination of melanoidins, increased the galectin-3 inhibition, and had the anti-cancer effect through increasing apoptosis, and of course it was structure dependent in the pectin. So what we did, we changed the molecular weight. So we modified the molecular weight of shallot, green papaya, and passion fruit, the albedo part. So in green and in green we have the native pectins, and in red and blue we have the modified pectins. So we decreased their molecular weight, and we saw that it had increased the anti-cancer effect. So the viability of the cells had decreased with no cytotoxic discovered. And we also saw that for these pectins, they induced the cell cycle arrest. So they stopped the cell cycle. That is one thing that the cancer cells has. What cancer cells have? They have the decrease in apoptosis and increase in cell cycle. So they are dividing really fast. Sorry about that, that is in Portuguese here, but we continue with our passion fruit pectin. It's we did the same extraction and modification of the citrus pectin that we developed in our lab, but now this time we treated some mice that were induced for colon cancer. But before that, we need to see if the pectins were entering the cells, and we saw that it was entering the cells in vitro. We also showed that we reduced the molecular weight of the pectins from here to here, so it was smaller and more homogeneous, and we saw that it had modulated the expression of proteins related to apoptosis and cell cycle. And in the in vivo with mice, what we saw, it was that the pectin, the modified pectin from passion fruit, it had decreased the number of pre-lesions and lesions of cancer. And the animals, they were feeling much better than the control that it was here. And this is the treatment of pectin. And the length of the colon was almost the same as the control. So we might have produced a new dietary supplement with some anti-cancer effect. And we continue doing it with the unripe and ripe fruit, but modifying it from the unripe and ripe papaya. This is also, I'm showing you that we could have a more homogeneous polysaccharide here. This is the control, the unripe and the ripe. And what we saw that we decreased the number of lesions in rat. It's two different models. We work also with rats and mice because depend of the model that we are using. So the papaya, we continues work with rat, and the passion fruit, we work with mice. And this is the spheroid panel that we saw that our modified pectins have decreased the tumor size, at least for the spheroid methodology. So it's a good perspective for application, just like the modified cytospectin, if we consider to extract and modify the pectins from shallot, green and ripe, unripe and ripe papaya, and also passion fruit. But remember, this is just a summary what we are doing in our lab. So we extract and isolate the pectins. We fully characterize the sugar component and the structure of the pectins, and we see some kind of biological effects in B-dimensional cells, three-dimensional cells, three-dimensional or B-dimensional organoids, and in vivo with rats and mice. So the next step is how low molecular weight pectins are absorbed and distributed throughout our organism. Is it commercial worth, or just eating ripe fruits could increase the anti-cancer benefits, or the modified pectins, they can be used as a cancer adjuvant treatment or prevention? These are all questions that we need to ask as our research is getting through these years. But I just brought some modified cytospectin that is regular in the market, in the US market, in the American supermarket. So this is the main focus of my work, is to create some dietary supplements highly insoluble pectins. But the thing is that, remember that I said we need to have a low molecular weight to increase their biological effect, but their technological effect, it needs to be high molecular weight polysaccharide. So another part of my research line, I am using now to create nanoparticles with pectins. But to create nanoparticles and to have some maybe anti-cancer effect or delivering some kind of biological or bioactive compounds, we need to put it inside a nanocapsule. So I work with this part of technological thing, that is creating nanoparticles with pectin, but with high molecular weight pectin. So what we do, we combine some kind of negatively charged polymer, that is the pectin, the de-esterified pectin, and we put it with the positively charged biopolymers. And then we use the protein that is called lysozyme, because they have so many positive charge depending on the pH. So what was our main question? It was if we can create some nanocapsules based on pectin to increase the bioavailability of one specifically bioactive compound, maybe two, but I will talk a little bit about the polyphenolic compounds, but also a galactine tree. Remember that I was talking about the galactine tree. So we have a beta galactose that it's a highly inhibitor of galactine 3, but if you ingest it, we are going to break it and it's not going to have their effect. So if we cover it in nanocapsules, we could deliver it to the target tissues. So what we did, we nano-encapsulated some galactine 3 inhibitors, and also we encapsulated some phenolic compounds, especially the anthocyanins extracted from blackberries. So this is just a photo of our nanoparticles. And as I said to you, we created this nanoparticle, but the important thing is that the nanoparticle needs to be a nanogel. Why a nanogel? Because our nano-encapsulated compound could be released throughout the digestion. So we don't want to inject these nanoparticles. We want to ingest the nanoparticle. So it needs to be stable in our stomach, and it needs to be released throughout our intestine. First, we have created this new proof of concept pectin and lysozyme nanocomplex to encapsulate the beta-lactose, that is a powerful galectin-3 inhibitor. But the thing is that if we take the beta-lactose, the lactase of our intestine will degrade it. So we created these nanoparticles, and we saw that it enters inside the cell in a time-dependent way. We also tried to create without lysozyme using some calcium salts, and we were able to use different kind of pectins with different degree of sterification. This work is not published yet, but my student he's finishing it. But I show you the beta lactose encapsulation. Coming back for the phenolic compound. So imagine that we had this anthocyanin that it's very stable below pH 3, 4, 5, but when it increases the pH, what is going to happen? This molecule will degrade. So when we ingest the anthocyanins throughout our gastrointestinal tract, it is getting degraded. So we can't have the best biological effects of the anthocyanins because it gets degraded. It's natural. So what we did, we encapsulated. And we did a lot of tests. So we put in different pH, different temperature, different proportion components. We elaborate these nanostructures with anthocyanins, trying to apply it to food products or maybe as a dietary food supplement. And what we saw some very nice nanocapsules. They are taken together because we dry it before the microelectron microscopy. And we managed to understand how the nanocapsules could release the anthocyanins throughout the intestinal, or it could increase their absorption just like as nanocapsule. And inside the cytoplasm, it could be degraded, the nanocapsule could be degraded, releasing our anthocyanin. And we saw that in a bidimensional way and three-dimensional way, our nanoparticles, they were getting inside the cells. So it was one clue that maybe throughout our digestion, the nanoparticles could be taken by our cells. But of course, we need to simulate the whole digestion process from the salivary fluid, the gastric fluid, and the intestinal fluid. So we managed this infogest approach that it's a very difficult methodology, but it's well respected in the scientific community because you need to put it with all the enzymes that our organism has, and we need to see if the anthocyanin would be released through the intestinal phase. And we saw here, I won't get too much in this graphic, but we saw that throughout the digestion, the anthocyanins would be released, but would be released in a not degraded way. Remember that in the intestinal part that we have a higher pH, maybe from 7 to 7.5, 7.6, 7.8, the anthocyanins, they will degrade. So our nanoparticle had protects the anthocyanin and had released it slowly. But we need to prove it in other methodology. So what we did, We radio-labeled our anthocyanin with technetium. We encapsulate it, nano-encapsulate it, and we give it to rats. And we, through a SPET scan, we saw where the anthocyanin, the labeled anthocyanin, without nano-encapsulation and with nano-encapsulation, they were throughout the digestive process of the rats. And here I'm showing you some images that we saw. This is the free anthocyanin, 30 minutes. Let me see, it's 30 minutes. Yeah, 30 minutes. Now, 1 hour after the ingestion. And we see that mostly of the anthocyanin free, it passed from the stomach to the small intestine. But the nanoparticle had stayed in the stomach. 6 hours later, we saw that the stomach was almost empty. The anthocyanins, the free anthocyanins, had passed through the small intestine and reached the large intestine. But the nanoparticles, the anthocyanin that were inside the nanoparticles, had stayed more time in stomach and had stayed more time in the small intestine. And 24 hours after, we couldn't see any anthocyanin here, but the nanoparticle were still in the stomach. We don't know why. We are processing this data, and we need to see why they are still in the stomach. This is the quantification of this image, so we see a lot of the anthocyanins that were in the nanoparticle inside the stomach. And this is the biodistribution experiment. So we made other experiments and we extract the organs. 24 hours of the nanoparticles were gave to the rats, and we see that there were a lot of labeled compounds. We don't know which compounds are, but we think that it's the fully anthocyanin. We saw a lot in kidneys and in bladder regarding that it had been absorbed, biotransformed, and excretado, delivery, and extracted by the urine. And we saw a lot in the bone. We don't know why. We saw in the bone, just in the nanoparticles. We are trying to understand why it is in the bone. But we are pretty sure that it might have been because of the bone marrow. We need to see it. This is just a summary of our experiment. So we label it and we gave it non-encapsulated and free. And we saw, this is a good graphic here, we saw the percentage of the total ingestion. You see that in the bone we have 0.07% in the bone of the nanoparticle. This is the nanoparticle that we don't know if what is inside these organs are the nanoparticle or are free anthocyanins. We can't say that. So what we saw is that the non-encapsulated anthocyanins, they had a very good controlled release in gastrointestinal tract, had improved the absorption, and had target to different tissues. This is my crew. These are all my students. Some of them are entering this year, next year. Some of them are leaving the next year, so I am really grateful to them. I passed the YouTube link, so they might be watching us. So hello students, thank you for the hard work. Yeah, I really need you. And this is my acknowledgement. So FAPESP, my university, my institute, we had money from CAPES, FORC, that is the center that I am affiliated to. It's Food Research Center, CNPq. I had my my collaborators in Argentina, Hong Kong, UK, Netherlands, United States, here in Universidade Católica del Mare, here Universidade Autônoma de Chile, and also University of Maryland. So thank you very much. I am open to questions. Thank you. Oh, you can find me in this QR code, enter in my website also. Let me see. Oh, 42 minutes. It's fine. Yeah. So do you need any questions, or we are going to discuss this after? Yeah, you can speak in Spanish. I can understand Spanish. I only can't speak Spanish. Thank you, thank you. It's not working. Vê agora. Acho que tens que aumentar o volume. Oh, my Spanish is getting better.
I can speak louder.
Okay.
Right.
Great.
My first question is: your modified pectin is based mainly on galacturonic acid, free acid, methylated acid, or a mixture of neutral sugars plus the galacturonic acid part?
It's a mixture because depending on the methodology that we modify it, we get rid of the methylation. So we need to be very careful because we know that the methylation part is very important for the biological effect. We don't want a pectin that it's zero in methylation. We want some kind of methylation. So if we use it, too many acid to modify it, or too many alkali to modify it, you will going to get rid of this methylation. So we need to be very careful. What the main it's because I can't put here because it's protected by a patent that we are asking, but we develop a very nice and green process just using temperature and ethanol. Just this. We can modify the pectin without adjusting the pH, without doing anything. Of course, we need to adjust the pH depending on the source, but we do it with we try to do it with without mineral acid, just with citric acid, and then we apply it high temperature, and then we do several washes with ethanol. So this is really green, and afterwards we can have maybe in the this whole process we can have from 80% to 90% of pectin in my dried sample.
So it means when you talk about pectin, in this case, that is mainly polygalacturonic acid.
Yeah, it's mainly polygalacturonic acid, yeah. But the thing is that we need to maintain the ramifications from the aminogalacturonan type 1 especially, because we have some galactans and we have arabinants that they are really potent but not isolated. They need to be attached to the polygalacturonic acid.
So it means that there will be maybe a main molecular way that will be more active.
Yes.
So there will be a range.
Yes, there is a range, and we had this range analyzed by the exclusion chromatography. So we had to, it's a quality control, so we need to extract it and we need to run the chromatography to see if we have below the molecular weight needs to be below 100 kilodaltons. It needs to be below 100 kilodaltons. The peak, it can be between 60 and 30 kilodaltons, but it can't be too low and it can be too high.
And in case you have different sources, so the structure of pectin maybe will be different.
Yeah.
So is there any main difference that you get from these results? Regarding structural activity.
Oh, yes. We need, first of all, we need to seek for the food the food matrix. We need to choose it. Before modifying it, we need to characterize it. So before we modified the pectin from papaya, we had to extract the whole bunch of pectins. We had to characterize it. We need to understand the esterification process and how many esterification, how is the percentage of esterification to develop the test, to develop the system. So if we want to modify passion fruit pectins, the modification process, it's a little bit different from the papaya, that it's a little bit different from chayote. I think this was your question, yeah? It's not the same modification. We need to understand our sample before the modification. Otherwise, we modify it too much and we don't have the results. So we have a very nice pipeline from after the modification, some very fast cellular challenges that we do with some cancer cells to see if we had at least a small decrease in the viability of the cells without being cytotoxic. So if we had it, okay, so we might have a new compound class that we can use from a new dietary source to create a new dietary supplement.
Joe, we have another question from Dr. Nicholas for you.
Okay.
Hi, very nice talk, very
Thank you.
stimulating.
Yeah, thank you.
And I have two questions. The first one is, you said us that at the end you found the signals in the bounds
Yeah.
in the several
The
hours.
radioactive
Yeah,
signals,
yeah. But you are not sure if it's only xanthocyanin or the nanoparticle. Are you thinking to make the same experiment with two different labeling or something like that?
Different labeling, I think we can't do, and I will answer you, because it's really difficult to label with technetium some compounds, because it's chemical labeling. It's a chemical. So since the anthocyanins, they get degraded, we develop a new methodology that no one has ever done, and we are trying to publish this article. We are going to send it on January, But the thing is that the problem is not the labeling. The problem is what we are going to find in the blood when we are going to do the pharmacokinetics.
But I think two kind of labeling, one for the xanthocyanin and the other one for the carrier.
Oh, yeah. We conducted I know what you're saying now. Yeah. We labeled the pectin. But we didn't run this study.
Okay.
It might be. But the thing is that the pectin, it gets transformed by our organism. We don't know if we are going to see the pectin inside the bone, if it is a nanoparticle or no. But I understand when you say, it can be a nice experiment to see what we are looking at it. It's the anthocyanin. Yeah, nice one. I will ask for my postdoc.
Okay. And the second one is, you said us that you have some data about the effect of the pectins on the microbiota.
Yeah, I have it.
And can you talk something?
I can. It's just because otherwise the presentation would be very large. So we have a model, in vitro model of colonic fermentation. So we get human feces from donation, and we have this pretty cool methodology of some kind of culture media, and we put our pectins, and what we see, we see the pH that get decreased through 24 hours of fermentation. We see the pressure of the flask, and we follow with the characterization of the short chain fat acids. And also we do the 16S sequencing. We were used to do the Illumina sequence, and now we are trying the mini ion sequence, and we are getting some good results. For us, it's fine to only to stay in the genus part, not the species. Just the genus, it's fine. And what happens? Our pectin, it is good, and it's good. Why? When you use apple pectin or maybe inulin, inulin, a fructooligosaccharides, or when you use lactulose, the fermentation is really quick. So in 4 to 8 hours, the pressure of the flask, it gets really high. So it's the same that if you ingest it, you will have bloating. It's something that no one wants to have in a dietary supplement. So our pectins, they get fermented slowly. So in the 8 hours, we don't see a change in the pH. You have a change in pH, but not too much. You don't see increase in the pressure of the flask, and you don't see the production of short-chain fat acids. But 12 hours after, you have it. 24 hours, you have. And another good thing is that it produces highly quantity of butyric acid that we know that it's had a very nice anti-cancer effect, and it stimulates the growth of the epithelium. So we have this methodology, but I didn't put it.
Thank you, Joao, and thank you, Nicolas and Gustavo, for your questions. We will be continuing with the talk of Professor Rajesh Sani from South Dakota School of Mines and Technology, United States. And if any of you have questions after the finishing of the talk, feel free to contact with them. But for the problem of time, we will be continuing with the second talk.
Good afternoon. Yeah, it's working. So, just to introduce myself, I'm Rajesh Sani, and I'm a professor in 2 departments, Department of Chemical and Biological Engineering, as well as Chemistry, Biology, Health Sciences in South Dakota School of Mines and Technology. So today I will talk about the extremophilic bioprocessing a little bit, and extremophiles. So Dr. Aparna, I would like to thank you organizers and Dr. Aparna Banerjee. Thank you so much for inviting me here, and I'm very excited to be here. I'm feeling much better than yesterday. I was a little bit tired. But today I'm very excited and very energetic, so hopefully I will do a good job today in my talk. But today I will be just focusing on polysaccharides. And I have a small overview. It's like today talk is totally different than the yesterday one. Only one slide I just took it from yesterday presentation. So today I will just talk about little bit about my group's research summary. It's only one slide which I took yesterday one. Rest are all the new slides. And Dr. Aparna Banerjee asked me to show some results or whatever we generated on Deception Island sample. So I will show those results. And then second story is Cynthia. She's sitting here. So she visited our labs. So I will show her results, whatever she did in our labs. And then I will talk about these polysaccharides and my group research on extremophilic polysaccharides. And then I will give an example. Dr. Fabi has done a very good job, especially for biomedical application of these polysaccharides, especially pectin. But I will show I will show a biomedical application for our EPS we got from a thermophilic microorganism. And then I will show some research facility we have at SD Mines, and in summary. So if you look at this slide, this shows the my group research summary. So like if I say exopolysaccharide work, it's like 5%. Maybe 5-6%, but rest of the 95% or 94% work is on extremophilic bioprocessing. So if I define my group research summary in one sentence, It's like microbial technologies in sustainable development of energy, environment, agriculture, and health. Means we use these microorganisms for various applications. For energy, we use for, sorry, it's too fast. Sorry. So for these extremophiles we use for advanced biofuel and value-added products, and then we also using those extremophiles for biomedical applications. So this is the something today I will talk about that little bit. And then a lot of work we have been doing using those microorganism and looking the rules of life of these microorganism or biofilm grown on various substrate. And then we also working on the space biology, because whenever you have extremophiles, you start connecting those with NASA. I mean, how the life is evolved and those sort of things. And then we also do a lot of genome editing with these microorganisms. I presented one example yesterday in my talk. And then we also using these extremophiles for agriculture applications. Like these microorganisms, they can produce bioplastic, which has the unique properties and we can use for agriculture application. And then one of the things we have been doing for last 7 years, because this climate change, greenhouse gas emission is like global problem. So we are trying to convert carbon dioxide, methane gas into value-added products. So let's start with my story number 1. First story is the Deception Island samples. Dr. Banerjee, Aparna Banerjee sent us some samples. So we we did some metagenomics. This is the, I'm not going to read everything, but this is the overview for metagenomics. We did it for these Deception Island samples. So we took those, we got about 11 samples. Then we had to pull those samples, make about 8 samples. And those 8 samples we try to do the metagenomics and mags. But I will show you we had some problems, like these samples, they had very low microbial load. So we had a very difficulty to find out the microorganisms or do the mags in with these samples. So these are the samples we got. We got total 8 samples, but we pooled 2-3 samples together. So these samples, and these were the temperature, and I think most of you familiar with those Deception Island sites. So these 8 samples, we try to, first step is we try to isolate the DNA. And you won't believe we had a hard time to find any DNA, means large quantity of DNA in those samples. So if you look at that, we use 3 types of kits: Zymo basic kits, Quaison kits. They are very famous one. You can extract DNA from any samples, but we were getting very low samples. Then we got a different kit, it's called like forensic genomic DNA extraction kits. Means you can extract very small amount, like nanograms of DNA from those from any samples. So we use these 3 kits, and if you look at the DNA concentration, it was very low. And most of the times, like, we had a hard time how should we proceed to do the sequencing for those samples. But anyway, we pulled those samples together. We started with the 8 sample and did the nanopore sequencing. So we used that kit SQK, this specific kit for these samples. So this flow cell, in one flow cell, you can generate about 30 gigabyte data. But because we had so low DNA, we could generate only 3.84 3.84 gigabyte data, only 3.84. And if you look at this graph, so like only this 3.84, like this only very small amount of this reads were qualified. Rest of they were failed, and they were not good reads. So we just got only very small data from these samples. But the N50 was like 1.5 kb. I don't know if you guys know N50 is the length of the shortest read. And total at the end, we just received only 9.28 megabyte data only. And you know that one genome, bacterial genome, is like 4 megabyte of data. So it's very low data. So what we did, we analyze all these data and then try to find out the microorganism present in these deception island samples. So then if you look at these are the 6 samples, and very interesting results we got. So what we did, most of the time people, they use only 200 nucleotide sequence cutoff, but we put the cutoff 500 nucleotides because we just wanted super accuracy. Accuracy means like whatever organism we are presenting, they are present there. So that was the case we were thinking. So if you look at this data, very interesting data we got. This fumarole, which is very high temperature data, very high temperature sample, we got only one hit, sulfitobacter. This is a very good microorganism. What does it do there? It degrade the algae, break down the algae, and generate the proteins or some food source for microorganism. And then we found that like this bacteria, especially sulfitobacter, it has been reported for, it has a unique lacase enzyme, and lacase enzyme can break down dyes or any organic pollutant. So this is the something I was talking to Dr. Banerjee, like you want to use those sample and isolate this bacteria. Maybe you can get unique lacase gene, and the enzymes, they have very big market if you get the good lacase. And then one of the most important thing I would like to point out, this photobacterium we found in all the 7 samples out of 8. So this was present everywhere, and this is a luminescent bacteria. It grow with, it has a symbiosis with the fish. So it grow with the fish, and I think it just take the food from fish and grow this bacteria, and fish use for luminescence. So this bacteria was present, and then we find several other bacteria, very interesting bacteria, especially this candidatus nitrosporulum, and some other like archaea type microorganism. And we did find some chloroplast, some hit in chloroplast, means there were a lot of microalgae present there. And then if you look at the next, so this bacteria was in 7 sample out of 8 sample. And the rest 2 samples, again you find this photobacterium was present in all the samples. But this sample, telephone bay, I think we found most of the microorganism in this sample. It's maybe because of the low temperature, and then we could see some like microbial DNA there, and we find several of microorganism. So we looked at all these microorganism, what they are doing somewhere, like they were oxidizing N2O, and they were doing lot of activity. So we have all the things, and there were some microorganism, I don't remember right now, but they were producing even hydrogen. So there is something now, we have a list of microorganisms present in those samples, so we can target and isolate those microorganisms. So what we did, and we also find some uncultured marine virus. Viruses, they were present in these samples. And we also find several reads for chloroplast origin. So they were probably the algae samples. So we looked at the literature. So far, but I looked at some results yesterday. I think Lacithia, she presented that she finds some marine microorganism, streptomyces, actinomyces type of microorganism. But these are the reported, these microorganisms have been isolated from the Deception Island. So what we did, we had a high school student and she tried to grow some samples and then try to, so what did it, she just plated those and got like about 30 microorganism. And before she left, she was a high school student, and she just put in refrigerator and she just went to her school. Then after 2 3 months, I asked one of my student, I have to make presentation in Chile, can you identify those bacteria so I can present something? So then my graduate student, she tried to grow all those 30 microorganisms and she could grow only 6 7. Rest of they did not grow. So I'm just giving you a message here. If you are isolating the microorganism from Deception Island, make sure you preserve them nicely. Because what happened, as soon as you put in refrigerator, something happened. Maybe they got the Rapid City, South Dakota air, and now they are not growing. And now those bacteria are not growing. Only 5 6 are growing. But the interesting thing is, she find one bacterium, uncultured bacterium. So this is something I am very interested. So I ask her, can you put in your nanopore and do the whole genome, and what is this bacteria is? And there are some like pathogenic type of microorganism. I don't know what they are doing in their Deception Island. Sometime I feel maybe she grew from our lab, these bacteria, but these are coming from the Deception Island. But this is something I want to give message. If you have those new cultures from the Deception Island or somewhere, make sure you preserve those before. Don't just keep on the plates, so they may not grow if you want to grow them again. So that was my story on Deception Island. In summary, we did see some unique microorganism present there, especially sulfitobacter, and that photobacterium is everywhere. And there were some microorganism which has an industrial application, so those could be isolated. Now, second story is, and I really want to say that one because Dr. Banerjee is going for sampling again. So make sure you take lot of sample or you do filter and you bring lot of biomass, because it's a oligotrophic environment. You don't have many microorganism or microbial load in those samples. So my second story is, I'm presenting your work. Cynthia visited us for like 2.5 months or less than 3 months, and she did a lot of work. She was working with my graduate student Priya. So she brought the 4 microorganism with her, 4 bacterium sample, and then she did the first thing she did the PCR and 16 sRNA for those sequences, and she found those they were bacillus type microorganism. And then she had like extracted the EPS for these 4 samples and did the SCM studies. And then I think she spent a lot of time to take those pictures and did some EDS. She was looking what type of elements are present, and then so she generated a bunch of data. And then this is the summary. I'm not going to show all the pictures. They are like hundreds of pictures she did and EDX. But here, these 4 type of materials, EPSs, and she found like elemental compositions and the morphology, the EPS morphology. Some were like porous and non-porous type, some irregular crystalline shape on the basis of XRD data. And then she also looked at some elemental composition. She found in some of the EPS they were sulfated. I mean, they had some sulfate, and these sulfated polysaccharides have been documented as showing strong antiviral activities. So if she start working on that EPS, so hopefully they will have some antiviral activities. And then the main goal of her study was she wanted to scale up, but it took us some time because the reactor was in my lab, and then we moved in different lab. But eventually she run this reactor, and she just run only one time. So she this is just growth curve and for that reactor, and this is the, I think, her serum bottle data, and this is the reactor data. So she could just do only one time, but these reactor things, you have to do several optimizations, and then you get the good results. So just to summarize, she learned several techniques, small techniques like DNA extraction, PCR, gel electrophoresis, purification, and she also got little bit experience on 40-liter reactor, and then some scanning, microscopy, and EDX, and she also worked on little bit NMR there. And she also made a presentation. We had a opening ceremony for a lab. So we just recently got the 260-liter reactor. It's a 300-liter reactor, so where we can scale up. And she made the poster presentation there. So if you have any questions on this, you can ask to Cynthia. Okay, so now my third story. So I talk about, because Aparna strictly asked me to focus only polysaccharides, so I'm just talking about only polysaccharides, and my polysaccharides work again 5-6% of my research work. So let's see how much I can talk. So right now I'm going to talk a little bit about the polysaccharides market. I thought this one something I just wanted to show this slide so it will be good for our discussion. And then I will talk about some work we did on thermophilic microorganism, bacteria for their EPS production. So if you look at the polysaccharide market, it's not bad. So here the 2022 is like about 100 billion something, and then is going up into 2032 with CAGR of 7%. So for me, if CAGR rate is more than 5%, is very, very good. It's a very good. And then if you look at that, this polysaccharides mainly coming from the plants like Dr. Fabi talked this morning. And I think his pectin is here somewhere. I think this is his pectin. So it's like increasing, demand is increasing, so you are in a good shape. But ours is also like, we are in others, Aparna, because our polysaccharides, we don't know, we have to name those, because they are not starch, they are not cellulose, they are not chitin, they are not pectin, they are not agar, they are not alginate, they are in others. So we have to name those. So I'm excited about that. And the good thing is, especially the EPS is coming from the extremophiles, they probably will have a unique properties. And I will show you some unique properties. So this is the very old slide I have one of my student Jia Wang prepared. So their applications, like, you just look at one of the application this morning, their biomaterial, cosmetics, biomedicine, but one of the problem is in their producing, like especially microbial production, limitations are most of these EPSs, they come from pathogenic strain, and they don't have the good bioactivities. And one of the important thing is, most of the microorganism you grow with sugars, like glucose and glucose, they are very expensive. If I compare the lignocellulosic biomass with glucose, like glucose is maybe more than $400 per ton, but lignocellulosic biomass like 10 times less, maybe $30-40 per ton. So can we use some other substrate to produce those biopolymers? So that's something that was one of our focus, and I will talk about that a little bit more in details. And this is for students. So if you look at the exopolysaccharides, you can produce from thermophilic microorganism. If you are looking for thermostable EPSs, you can produce from psychrophiles, halophiles. There are a lot of results. There are a lot of reports from halophiles, acidophiles, and alkylophiles. And then our focus has been on thermophile, and EPS has been produced by mainly Bacillus type and Rhodothermus and Thermus species. But if you look at that here, most of the microorganism we are growing with expensive sugars. And you know, this food is like more than 20-30% cost to the process, because this is so expensive food for these microorganism to produce the EPS. So we thought, why we cannot use the lignocellulosic biomass, which is, you know, abundantly available throughout the world, and reduce the cost of their production. So we started using the lignocellulosic biomass. But as soon as you start using lignocellulosic biomass, you know, one of the thing is you have to do pretreatment, you have to do hydrolysis, and then you do the fermentation. So one of the idea I think I showed yesterday, one of the idea is, can we find a extremophile which will do everything, like which will do the pretreatment, which will do the hydrolysis, and which will do the fermentation and produce the EPS. So we did find that one of the one thermophilic microorganism. And then we can also use some other feedstock like natural gas. Natural gas is mainly composed of the methane gas, and which is it's also inexpensive compared to the sugars. So, but our focus has been on lignocellulosic materials, where you can get the lignocellulosic materials like less than $40 per ton compared to the $400 ton glucose. And then you have to do pre-treatment, you do the hydrolysis, and then produce the EPS. But what we have done, we have combined all these steps in one step, and we say new form of consolidated bioprocessing. So what we did, I think while ago, we did this isolation of microorganism, and we found a bacterium. It's different than the PHA producing bacteria. It's a Geobacillus species, WSUCF1. So this bacteria can grow untreated corn stover and produce the EPS. Most of the thermophile, you know, EPS is like, you know, a little bit stress, you know, they produce in under stress conditions like heat stress. So most of the thermophile, they should produce EPS, but their yield may be little bit less. So we use this bacteria, and then first thing what we did, so we grow this bacteria on a different carbon source, and then we found that this bacteria was producing EPS with tons of carbon source, with mannose, glucose, xylose, means pentose and hexose sugars, even disaccharide it was producing. And then we map all these pathways in this microorganism, because we have the whole genome of this microorganism. So we map, and then we got the production of this the EPS nucleotides. Basically, they are the precursor for the EPS. And here is the just I'm not going to show all the flask level or reactor data, but this is the reactor data. So this is the EPS production. This bacteria is so nice, like, you know, within 8 hours it was producing the maximum EPS and growing. And then what we did, we precipitated out that EPS exopolysaccharide, and then use the gel filtration chromatography and purify the EPS. We found 4 EPS, they were like very low, so we did not characterize those, and we characterized those 2 EPSs, EPS1 and EPS2. And this one were very low concentration, so we did not characterize. And this, again, these 2 EPSs are coming when the bacteria was growing with glucose. And then we also did their chemical characterization, like, you know, the carbohydrate contents and nucleic acid, whatever they had, like their composition, chemical composition analysis. And then we also did some NMR and FTIR things, and then we found the little bit structure, primary structure of this EPS. But we believe that these EPS, they have a secondary structures too. We still did not elucidate those structures. Then here we try to compare our EPSs with reported literature. So one of the interesting thing is that we found that the our EPSs were, they were mannose and glucose, and EPS2 were mannose. And the EPS, which was composed of mannose and glucose, it was so highly thermostable, like the degradation temperature was 319 degrees centigrade. Very, very thermostable EPS. Means this can use, you can make even autoclavable drug delivery system using that EPS, because 390 degrees centigrade is a very high temperature for this one. But the second one, which has only mannose sugar, it has about like 314 degrees centigrade the degradation temperature. So now again, this was just our control, but then we grew this bacteria with corn stover. That was our main focus. So we take the corn stover, untreated corn stover. I'm sorry, my mic was away from my mouth. So we take the corn stover and grow this microorganism, and then we grow in this reactor. And here, in this case, when we purify the EPS, so we found 4 EPSs, EPS 1, 2, 3, 4, and then their composition like similar to glucose, mannose glucose, mannose glucose, and mannose. And then we looked at their degradation temperature. So their degradation temperature, like, again 318, but some mannose glucose, they were like 200, 226. And then we also look at the little bit, like Dr. Fabi did a lot of work on this non-cytotoxicity, but we also look at their toxicity level. So they were not toxic to the human kidney cells, like up to 2000 means is like 2 milligram per liter. They were not toxic. Now these EPSs, like, they are very thermostable, they are very less, they are almost non-toxic. So one of my student, like, he was very interested in biomedical engineering. I'm not the biomedical person, but he was very interested. He said, I want to see if I can use these EPS as a drug delivery system. I said, okay, go ahead and use it. So then he used these EPSs for a couple of things, like, for antibiotics delivery as well as anti-cancer delivery. So first is, like, what he did, he tried to make the film for this EPS, and then use its staphylococcus and look at if this film is killing the staphylococcus pathogen. So he used the antibiotics amikacin and delivered to the pathogenic cells, and this was very effective. So we already have published that paper. The second one, this is, I'm going to show a little bit more detail. So he again prepared a film and functionalized this film with the this anti-cancer 5-FU, and then study with the anti-cancer cell. Okay, so I'm not going to show those results. And why we are using EPS, just to show that traditionally we use PLA, PEG, or PLGA. So they all have their limitations, like PLA produce some acids, and the PEG or PLGA, they may produce, like, 1,4-dioxane, which is very toxic to the cells. So we use our thermophilic EPS for this drug delivery system. So benefits for this, exopolysaccharides yield near almost zero toxicity, or they are very non-toxic. And because these exopolysaccharides, they can be degraded by, you guys saw in the morning, they can be degraded by our enzymes. So it's safe. and they have resistance toward the hydrolysis at various pH and temperature, anti-malignant tumor behavior, West majority, so many benefits using the EPS. So then what he did, he isolated the EPS, but he did not purify, because purification is a, again, add to the process. So we started with the crude EPS, and he make the film. I don't know how to run that film, if we can press, but so this is a, like, he isolated the EPS from, yeah, thank you so much. This is much better. Yeah. So he prepared that, and it's like, very flexible and good property. And then he functionalized this film with the this anti-cancer 5-FU, and treated with the cancer cell, and it was effective. I won't say it was too effective, but it was effective. But the idea was, like, if we can functionalize this anti-cancer, and especially it's like, he wanted to do through skin delivery in through the skin, so we can put it there, and it will release the anti-cancer slowly and kill the cancer cell. So that was the idea. So it's just showing the data, like, how this film without anti-cancer, and with this with sorry, without and with. So he could see few bands, like he could see that there is a functionalization of this 5-FU to the film. And then he treated this film with the human melanoma cells. I don't know about these cells, but they are cancer cells. So he treated with that, and look at the there is a, in biomedical, there is a test called MTT test, where you look at the cell viability. So as soon as the cell came in contact with this film, they instantly died. Okay, you can see quick died. But you could make the, maybe he used the 5%, so maybe you could use 2 or 3%, and then probably they will die slowly. But this film functionalized with with 5-FU. It was very effective to the cancer cells. And then he also did some this fluorescent microscopy. The main objective was to look at if there is a difference in the cell morphology, or how the cells are dying. So he could not see, like, this is this is a control. After 12 hours, 24 hours, some cell disappear, means the total cell fluorescence was decreasing, but it seems like it was not like, cell were getting deformed, or they were getting, like, changing in the cell. But he could see the difference in the total cell fluorescence. So, and then he also looked at the release of this anti-cancer from this film. So there was like burst release in the beginning in first 3 hours. Then after 3 hours, it was very controlled release. And that's something we want to treat the cancer cell. So in summary, we could make the film. That's a very important thing. These are, exopolysaccharide. They are soluble. You know that they are very soluble, but he could find out the cross linker and some other things. He could make the flexible film, and he could functionalize that film with the anti-cancer, and that film was very effective to the melanoma cells. So this already we have published that paper. So that was my third story. Now I think I can finish in few minutes. So I'm not going to show the research facility as School of Mines. I did not put all the research facility, but this is our lab, my group lab. So where we have this meta-omics lab, we say. So we have all the sequencing facility, and we also provide students training. I hope Cynthia see that CBAC 200. So that's a CBAC 200 lab. And then we also have the anaerobic chamber, confocal microscope, and we also have single cell genomics. So we have all the things. So the idea behind this, it's like, it's a nanopore, it's not that expensive, but the idea was like, we wanted to teach our student how to do sequencing, how to play with the big data, so they can do stepwise. Like most of the time what we do in university, we just isolate the DNA and RNA, whatever, and ship to the company, and then we don't know what's going on. But I wanted my student to get frustrated how to prepare the library, how to check the DNA quality, and then how to do the barcoding, and then how to do the sequencing, and then when you get 30-40 gigabyte of data, how to deal that with that data. So that is something I want to, and it's working very nicely with my students. So this is our single cell genomics. So but this is a very expensive things, like we, like for example, if you want to do the single cell genomics for one microorganism, you have to design more than 30-40,000 probes, and it cost, designing those many probes, it cost about $20,000. So if you want to do single cell genomics for one microorganism, it's a very, very expensive, but you get very nice good publication at the end. Okay. And then we do have a reactor. They are in different lab now because we are shifting here and there. So we have a 3-liter reactor, we have 40-liter reactor, and then we just got 260-liter reactor, and this is the 3,000 liter. It's in different city in Brookings. So I went there and I took my picture to show that we have 3,000-liter reactor. So we can, like, we can scale up. If you have any process at flask level, come to School of Mines, and we'll scale up to 3,000 with discount rate. Okay. And then this is just to show you how these reactors, we are growing. We have a research scientist, Dr. Ankan Sinha. He's a process engineer, and then he does a lot of our fermentation work in our group. And then we have a Cape lab. You saw that Cape lab. We can do anything with polymer. If you have any polymer biomaterials, so we can do TGA, DSC, and they have so many equipment. I'm not the polymer scientist, but we can do anything with the polymer there in that lab. And we recently got a one equipment is called Nano IR3, and this is I won this award. I'm a PI for this. It's a $800,000 instrument, and it's ready. If you want to do something, I think Cynthia is doing. We have 4 samples. So right now we are generating SOP for this equipment, and this is me here. So it's a like, FTIR. FTIR resolution is like 1,000 micron, but this guy can go up to 10 nanometer. So it does the AFM, and then you can probe a single wavelength and find that peak. So like for example, I have a bacteria, and in bacteria they in bacteria they produce like PHA or bioplastic. So suppose I can take a picture like 50 by 50 micron. In that 50 by 50 micron, I may have 200 to 250 bacteria. And then I don't know which bacteria has a PHA. So I can scan, and that can tell you out of 200 bacteria, your 5 or 10 bacteria has PHA, rest they don't. So you can do even quantification in native stage. And then this machine also has you can also do thermal analysis. So this is a very nice equipment. This in my lab. If someone wants to use, you are welcome to use. So in summary, it's not the scientific summary, but because Aparna asked me to talk about the polysaccharides. So, but Deception Island, just to summarize this, Deception Island new isolate shows like, they should be tested for biopolymers. In fact, we should isolate those new microorganisms from the deception samples. And then I told about the extremophiles. These extremophiles, they have a thermostable EPS, and those could be used for biomedical applications or other applications. And we could use agri waste as a feedstock for those thermophilic microorganisms. And using the agri waste, especially in South Dakota, it can strengthen our agriculture economy in our state. So that's a very important thing, and promote the economic growth of South Dakota and eventually among the other states in the United States. And then last message is feel free to use your facility, send your students in our lab, metaomics or productions of biopolymers or other things. And then I would like to acknowledge the graduate student Jasmeet. She helped the high school student and isolate those microorganisms from Deception Island. Priya was mentoring Cynthia, and Dr. Ram Singh did the metagenomics study. And then I have a very simple cartoon here, and it will say something. See, stop on me. Thank you so much. Any questions for me? Sure, please. I have extra slide too, my goodness. I'm not going to show those. Yes, please.
Dr. Sunny, thank you very much for your presentation.
Thank you.
It's really nice. So, it's just a question about the polysaccharide. So, you said that two fractions of the polysaccharides. I remember you put in a gel permeation. You show it that they have 1,000 kilodaltons.
That's a very good. You caught it. I think when we purify those, so we did
Yeah.
not have the GPC that time.
Okay.
So like my student, he just used the G200 gel chromatography,
Mhm.
and with the marker, so he was getting they were coming so quickly because they are high molecular weight.
Yeah.
They are very high molecular weight. They are not 1,000. So
No,
they,
it's more than 1,000.
than
Yeah,
1,000,
because
yeah.
it's your void
Yeah.
volume. It was before void volume.
yeah,
Yeah.
good catch, because this is the incomplete data.
No. But,
Um,
but it's really nice because as you said as you saw in my presentation,
Yeah.
the high molecular weight polysaccharides, at least with the one that I work, they are not really soluble. But this is quite
These are
interesting
soluble.
because
These are
they are soluble.
These
Yeah,
are
they're
soluble in
soluble.
water. Yes.
And it might be because of maybe they are charged, maybe sulfated, or it's fantastic. It's
EPSs, they are they have lot
yeah,
of
it's
functional
really
group.
nice. Yeah.
They have lot of functional
Yeah, we
group.
saw some in
Yeah.
our laboratory
Yeah.
from Aparna,
Yeah.
but some of them they are low molecular weight, but some of them they are high molecular weight. Yeah, two fractions. Yeah.
So what we did, like that's a very good. I'm sorry, I skipped those things. But we grew this bacteria with two carbon source, one glucose as a control, and corn stover. So when we grew with glucose, so then after 24 hours or whatever, student precipitated those EPSs and then purify. So during the purification, he found two peaks. So there were two different EPSs,
Mm-hmm.
and then we characterize those. But in case of corn stover, he got four peaks. So there were four EPS from corn stover, because corn stover is a complex sugar.
Mm-hmm.
Sugar, like they were using in the beginning, like they were using some hexo sugars or
Yeah.
pentose sugars, or so we got four peaks and we characterized those. But Jia Wang, my student, he was working like 4 5 years ago. So after that, we did not. No,
Mhm.
I don't have anyone to purify and do the right characterization. So we just
And it's very interesting because most of the microorganisms that we know, well, I know probiotics, this kind of thing, they don't produce exopolysaccharides. Well, they produce it, but it's not to protect them. It's just because they produce it. And even if you change the carbon source or the culture media, they produce just a little bit. And this is quite interesting because this kind of microorganisms, as they adapted their cells really quick. It's something amazing. So it's thinking about the biotechnological approach. You can start changing the carbon source and the nitrogen source to modulate the exopolysaccharide production.
Yeah.
It's fantastic. Yeah.
So I don't have that slide, but like we, it has all the machinery for EPS production. And then most of the time when we grow with any substrate, they produce EPSs.
Yeah.
Yeah.
And it makes sense because it's a microorganism that it lives in extreme conditions, so it needs to protect themselves.
Protect themselves. Yeah.
From high temperature or low temperature.
Yeah.
Great. Thank you.
Yeah.
We have another question for you from.
Sure. Please.
Hi, Professor.
Hi.
Very interesting talk
Thank you.
and quite interesting results as well. Um, can you show the picture of the where you isolate the bacterial from Antarctic, please?
Oh, sure. I think yeah, that's I think in the beginning, hopefully. Yeah.
Yeah, this is one. This is one. It is quite interesting to remark that some bacterial found, for example, Azospiridium brasilense and Bradyrhizobium, those bacteria are commonly found in soil. So forming some kind of association with plants to promoting nitrogen absorption
That's right. Yeah.
from them. So I don't know if Aparna could mention that if there is any plant around there surrounding this part where they were isolated.
Mm-hmm. Okay. Mm-hmm. That's right. Yeah. Yeah. Mm-hmm.
Well, this is interesting anyway
Yeah.
because if you say that this site temperature is 55 Celsius degree, so these bacteria were able to live under this condition. Maybe they suffer some kind of mutations that allow them to live there,
Yeah.
even when they
Yeah.
come from the continent.
Yeah.
So they just settle down in
Yeah.
this place.
Can I add something? Like on your next sampling trip, maybe you want to do sampling where there is no human activities. So that probably may give you the microorganism present in the deception. Okay. 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 Mm-hmm.
And regarding these results also, do you have any advice regarding sample processing? Maybe sample processing must be do in situ in the base. I don't know if this is possible.
That's a very
Aparna.
good question. Like in our case, like as soon as we got the sample, we were very careful, and that was the reason. Like we use totally different criteria. Like in literature, people they use only 200 nucleotide, but our cutoff was 500 nucleotide. So we just wanted to make sure whatever match or hit we get, it's coming from deception ion and not just matching something. So we use the super accuracy, but the only problem was like the DNA load, microbial load was so low. It took us months and months to isolate only DNA.
And
And
often 5, 6, 7 time that they
we
you have run it
isolated nine times
9
and then
times.
pulled that DNA
Hmm.
and did the metagenomics.
No, we were talking like last 2 days, we were talking how to improve our sampling, and this time Alexander and I we will be going to collect samples, but we will be taking care of Alex because now I have talked with him and we know that what is the optimum way to, and also to increase the quantity of sample and not 50 grams. That also we were talking. Nicholas also have a question for you.
Sure, please.
Thank you for your
Thank
talk.
you.
2 point. I think one comments thinking that we are thinking about a new project.
Sure.
Uh, Maybe the solubility could be a very interesting point
Very
to
nice point. Yeah.
to test
Mhm.
if depending of the cells, plants of microorganisms,
Mhm.
and because of the biotechnology application,
Mhm.
it seems that it's a very good point to focus on. So just to take on the idea, and just I'm dealing with extremophile bacteria, acidophile, and we are capable to induce biofilm formations using homocysteine lactone molecules that are involved in cells communication. So we can also explore this. I think it will be interesting to test your models
Sure.
about
Sure.
chrome sensing,
Sure.
and I'm very interesting to send you some samples to grow at the very high scale. With your last um, instruments,
Mhm.
is it possible to see or to measure extracellular vesicles?
That's a yes. It could be done. It's I think this Nano IR3 has a lot of applications, and just to let you know, my group has been focusing on biofilm.
Yeah.
We do those. We working on the SRB, sulfur reducing bacteria, especially the corrosion thing. Uh, how can we minimize the corrosion?
Okay.
Can we do the 2D material coating? So we are looking if we do the 2D material coating, what genes are expressing. So more, but it can be measured. I think nano IR3, you can go nanoscale level. But the only thing, like, if we know in vesicles any peak or wavelength, we can take a picture and we can scan. Yes.
Okay.
This is a like new equipment for us, but like we'll be learning a lot of things, especially in
Because
life cycle. Yeah,
we are capable to extract, to purify the extra vesicles.
I think so.
So I can.
If we know, like, some peak, this is the wavelength, like this is the particular peak for protein or lipids or polysaccharides, or we can just scan, and if we find that peak, it means that is present there.
And the last point regarding the other polysaccharide, this is a good description, and we have one in acidophile. His name is pale exopolysaccharides, and it's just growing at the interface between liquids and hair. And I think and it is present in all the acidophile that we can found in the database. So maybe we have another point to work for the project.
Sure.
Actually, the idea is that I
And,
will be.
yeah,
Yes.
means like in whole state there is no machine like that, so this is the only in my lab that machine we just got a few months ago. Now we just will be playing with this machine, yeah, and ready to help.
Thank you so much, Professor Sani, for the extremely nice talk. And we have a lot of students, so for the students, it will be it, it will be a very good learning moment to the next one. So we will be going to the next phase of today's event. We will be going to a round table talk to talk more about the polysaccharides and the scope of polysaccharide research here. If we can go to the presentation, please. So I will be in brief. We will be talking about the points, because in this project we had some mention. There are people that won this year, for example this fund. Daniel is here, and Alex, who got before me this fund. So this fund is a very interesting fund in Chile, where the project is focused into outreach, and the outreach of regional institute with international institute. So that's a very good point. Have few modes, mode A, mode B, mode C, mode D. In the mode A, we expect to send some human resource formation from Chile to the outside institutes, the foreign institute. In mode B, we are expected to have some foreign researchers to come to Chile and get working here, that we didn't apply it for. In mode C, it's about vinculation or outreach among the international and national research institute, establishing some seminar, establishing some workshop like this, that we already had a national workshop before, where we had all the co-investigators of the project presented. Cynthia was presenting that time online from States, and this point D, mode D, is about accessing the scientific equipments of the foreign institute, that I was for a time in University of Sao Paulo in Dr. Fabi's lab, and Cynthia have accessed some of the equipment in States in SDSMT. So in all the objective that we have promised, it's a really small time project, 1 year. So it's like we get the fund and we run to work on the fund, but in 1 year we have finished all the objectives that we promised, and we have finished more than that actually. So in brief, I will be mentioning that, as promised, according to the mode A, Cynthia, the doctoral student in translational biotechnology in UCM, she was there for 3 months in South Dakota School of Mines and Technology, and I'm sure this will be helping her formation of as a researcher in future. And she has definitely learned a lot more techniques there that she was having here. So that's a very good point. Dr. Sani is also co-investigator of our INACH regular project of Instituto Antártico Chileno. So we'll be continuing the work of Deception Island polyextremophiles and their polysaccharides, where Catherine is working as a thesis student. So probably in future we will have more collaborations, including her. There have been manuscript communicated, one manuscript to be communicated. The collaboration is continuing. This is also to mention that the alternate director of this proposal, Dr. Galan, we are also co-investigator of INACH regular project, and we will be sampling this year also, and the collaboration will continue. Also, as promised, according to mode D, I have been into Sao Paulo, and we have characterized a number of polysaccharides. The same we have done will be continuing in Fondesit regular. In Fondesit regular, also in this project, we have foreign collaborator Dr. Fabi, so we'll be continuing the collaboration. There have been 2 publications. Dr. Shorkar, she was included in this proposal as postdoctoral researcher, and one is already published and one is accepted among us. And we will be focusing into design and development of advanced bio and nanomaterials. So this year we have on another FOBI project, where our co-investigator Dr. Esteban Duran from Universidad de Talca, he on it, and I and Adolfo will be continuing there as co-investigators. So that is also, we are continuing the collaboration. And we have incorporated a new doctoral student, Dr. Srijan Banerjee, who is already started working with Dr. Duran in University of Talca in the bioactive compounds PhD program, and he's also working with me. So the idea is all the proposal, and also one of the idea was to incorporate and open the network to others. So in the last workshop, we had already listened the presentation of Dr. Karem from University of Antofagasta. So we are also trying, we have opened the in-network biological polysaccharide, and we'll be continuing working on it. There is possibility to reapply the fund, that we will try. There is possibility to apply some more funds, that also we can continue. The next one, please. So just to finish up, these are some of the events that have been done about the publication, the collaboration. One of the point of this kind of project outreach, that we are having the media outreach also, and I'm really thankful to the Universidad Católica del Maule for giving us all the opportunity. In last 1 year, we know that we have been pushing for some of the events, because it's given 1 year. It's actually not even 1 year, because the fund arrived in January and we are closing the project in December. So it's really small time to do it all, but I'm really thankful to all the co-investigators. With few people I was working for last 5 years, like Dr. Gustavo Cabrera, and I'm really immensely thankful for all the help. Gustavo is a very like expertized person in polysaccharide and biopolymers, who have years of experience. Then with Dr. Galan, we have been started collaborating into the INACH regular project, and he has long experience in environmental characterization. And we had someday one idea that the polysaccharide production in the microorganism, or even the plants, but not the higher plants, there is impact of environment. And how about having an interdisciplinary approach to start understanding how the extremophiles and how the microorganisms are producing the polysaccharide implemented with the environmental factor. And that really functioned well, because we continuously went INACH regular and then Fondesit regular with this interdisciplinary approach putting on. And then there is Nicholas. With Nicholas also we started thinking one day, because Nicholas for a long time working on pale exopolysaccharide, and because I am a polysaccharide enthusiast, whenever I see someone working on polysaccharide, I go to them and I try to talk to them and incorporate them in. And some of my students from the lab are also here, so they are continuing working on polysaccharide. Adolfo worked in something very interesting, that is using the polymers for water treatment, that very few people are working. Srobona's PhD topic was also that, using polysaccharides and nanoparticles for wastewater treatment, that we must focus on and we should continue working for. So we will be calling all the co-investigators of the project. If we can please come here, and we can I will be keeping a simple question to all of you, that what is the expectation from all of you? How is the future of polysaccharide research in Chilean context, national context, and in the context of South America? And what is our next goal, and how we are seeing in future to continuing the collaboration? So if we can have please Gustavo, Nicolas, Joao, Alex, Professor Sani, Adolfo, please. Srobona, Cynthia. Yes.
Mr. Gustavo, tudo bom?
Professor Sani, finally we are nice to meet you.
Maybe we'll be starting with Alex from the environment.
Oh. So thank you, Berna, for having us here. And I mean, for me, it's a really new world, this of polysaccharides aspects. It's not my field. So from my perspective, I guess it's a really big field that we can explore, especially from extremophiles, because this is the theme that we are working on. So I guess this is really, all of you working in different lines of this production of polysaccharides and functions. So I guess we can explore more environments. Aparna is working not just in the Antarctic, but also in the other hot springs. So this is we have a really special place here in Chile, because we have all of these environments, different environments. So I guess the projection of these kind of studies is really big. So I'm really happy to participate of this network and try to understand how the environment, regulate of the production of these polysaccharides. So.
Thank you, Alex. Maybe we can hear to Nicholas, because Nicholas is focusing into the synthesis of polysaccharides.
Thank you, Aparna. Thank you to to create this network. I think it's a really, really nice experience. And I think that we have a very big challenge, multidisciplinary challenge, following to working together. And we have to think about extremophiles condition, or space, or environment, but we have also to think very close, not only environment. There are polysaccharides everywhere, I think. And I really hope that we can apply for new grants together from colleague from Chile, but also with from Brazil or from Dakota states. And I will help, or I want to promote the idea that we can manipulate microorganisms to produce enough quantity of exopolysaccharide with specific application, and this is what I want to do in this rate.
Thank you so much, Nicolas. Adolfo.
Well, in first place, thank you, Abarna, about the creation of this network. I think there are very big opportunities to work with bacterial polysaccharides, and there is a big opportunity here in terms of that network, the people involved in this network, to prove a lot of things. In my case, I work with the capture of heavy metals and organic pollutants. So besides the big opportunities in the medical field, the other hand is to prove these bacterial polysaccharides as environmental opportunities to remediation of water in my case. Thank you.
Thank you, Adolfo. For Cynthia, maybe we can hear her experience, how it was officially today, and what she expects in future from the network.
Hi, thank you so much, Dr. Aparna, for including me in this project. For me, was a process of learning too much. I learned too much. I am biotechnology of profession, so I finished my career and started my PhD grade in the same time. So I don't have the opportunity to work too much in lab. So I learned too much about the bioreactor. I go with my sample for the reactor. I say, here's my sample. So what you gonna do? I say, I wanna use the reactor. No, you had to learn a lot of things after to use this. So for me, it was an amazing experience. Go to South Dakota. Thank you so much, Dr. Shani, for receiving me. And that's my experience there. And sorry for my English. I'm learning about this. The what? Birthday parties. The parties. Yeah, they have in the lab celebrate all the birthdays and all the was an amazing group there. Received me with happily, and no, it's amazing the experience. I enjoy and I learned too much, and I hope go again and work again there. Yeah.
To talk about Cynthia. With Cynthia, I'm working for last 3 years. She has been with me in the in her undergrad trabajo de grado, then thesis, and then she entered into the doctoral program. And when she came to me, she has the kid of 9 months. And for a moment, I have not seen Cynthia giving excuses of the kids. Like she presented the topic so simply that Professor, my kid was having fever today, just like that, putting the points on the table perfectly. And often we think, we talk about, we have the gender parity groups, equality groups in Chile that we often talk about, but she's a perfect example of someone who never put the points that she's a mom, and she finished on time, she didn't took a year extra, she entered the doctorate, and she's going so good. Like if I as a tutor of her, that I'm not really her tutor now, I'm co-tutor, Alex is there, her tutor directly, and Alex will have an amazing student. She's so perfect and so particular and punctual to the work that we all must learn from Cynthia.
Yeah, I go alone to use Africa with my son, 3 years old have. So was a little difficult first, but with the help of the people there, give me all the help for my son and be working there with. Yeah.
And that was 9 months, now 3 years, by the way. The
Yeah.
kid grows. Gustavo, please.
I think that all of us have in common that we want to, through polysaccharide chemistry, to improve human life. So and now we are starting from microorganisms, from plants, from fungi or algae or something like that. But we have in common that we have to improve human life. That's why we are studying, we are doing basic studies and applying studies, and to try to connect the benefits of polysaccharides with human health, and also with human contamination, to improve air pollution and things like that. Indeed, microorganisms are everywhere. So this is crazy. It is expected, and indeed, thermophilic microorganisms may have an unique physiological machinery. So that's why they are able to produce interesting polysaccharides. Right now, Aparna, just one microorganism was able to grow up in aerobic and anaerobic condition as well. And under this condition, they are producing different polysaccharides. So it is a big step forward. So I never found this before on literature. So it is huge. And this is made like people like us, regular professors at universities. So for students, this is some kind of encouraging to follow this pathway that we are just walking together with them, by the way, because they are not, they are our working force. So help us a lot at the lab, presentations and so on. So I think that right now we have several results that will be published and have been presented in some congress and so on. So I think that next step is to try to set up another project to try to reinforce and to strengthen and to widen the network, because now we are just getting knowledge and starting, and I think that we are in a good fit to do that. By the way, the project that you are trying to set up now, it is an interesting one project. So okay, that's all.
Professor Sani.
I think I already said so many things, but I would like to thank you to Aparna leading this effort. I think the main thing is, or difficult task is putting together people. I think this is something very exciting. Thank you, Aparna, for doing this. And I think we do need sustainability. So I have few idea. I don't know if you will like it or not. But first, let's register this biopol, biopolymer, and I think we could say polysaccharide is very narrow. If we could say just biopolymer, so we can put some more people on the board. And then I would suggest maybe in next few months or something, of course, to sustain this biopol, of course, we have to write the proposal so we can meet again together. but I would suggest let's organize a small conference, like international conference of biopolymer, where if we can apply some funding and then invite more people, maybe 2 days conference, and then I know it's a little bit work, but it may pay off a lot.
Actually possible with the reapplying with the project.
Yeah.
It's a very good idea that you was telling.
Yeah, and then invite students, and maybe at the end, like we have a speaker, but at the end, like a specific workshop, so students can get some hands-on, and hands-on means like they can get really good notes on biopolymer, like biomedical research or bioprocessing research, and I think that's something can be done, or like workshop, biopolymer workshop. So, well, yeah, and then short course type. So this way, like we can include a lot of students in our. But first thing is, let's register this, and I really would like to take one picture with this group. And okay, third thing is, I think let's look at the opportunity where we can write the proposal and work together. As I was talking to Aparna, like if you are doing alone, you cannot go far.
No.
But if we work together, I think we can go really far. Yeah, I think that's it I want to say. But let's sustain it.
Yes.
I'm very excited now about this polysaccharides things.
Just one thing, just a little bit that one of the main challenge to polysaccharides to try to bring them to the market. Fortunately, we have good examples that it can be done.
Yeah.
So gel and gum, santan gum, and so it is possible. So I think that we are on the right way.
Yeah, I think you got a very good point. Sorry to interrupt you, but got a very good point. Like Dr. Fabi is working on pectin, so he knows what pectin looks like. But like we are working EPS. We don't know what are those. We don't know their structure. But the thing is like we can do some systematic study, like isolate the microorganism, first characterize those EPSs, and then look at the suitable applications. So we can move in food industry or environmental bioremediation things. We can use those EPS, or we can use those EPS for biomedical applications. Maybe with more good activity, antiviral activity or antimicrobial activity, we can do. It's your turn now.
First of all, thank you, Dr. Aparna, to incorporate in this group. So today's talk, all are talking. So here, all there is someone who is extremely expertise in the augmentation of the production, like Dr. Sani. Dr. Gustav, who is know how to take the something to market. So I think we can sustain the group to start from the basic, like the scratching, how the factor is affecting the production, and then start that to up to the marketize. We have the post.
Before I forget, so one thing, we can also put industry people on board on this.
Yes, that was I want to tell.
If they can support these activities, not only funding agencies. So this way, I can come again in Chile.
We need that.
Yes. So I think the final future that can be again funding, and from this, not in 1 year, 2 year, but after 5 years we can probably produce one center focused on the biopolymer.
The another thing is, many students are here, and feel free to contact to any of them, and moreover all the people that you are seeing here all have funds. So like we need more students definitely. Some are
Not in
here.
one application, in different application, who have the interest in
Yes.
biomedical person is there, who have application interest in food industry person is there. So different aspects is there. So if we can organize one center, a little center start, then students can come to learn on course or like this, and then start funding more. That's my idea.
Students are our workforce. I understand we have break here, but I think we should organize these things. when we have full-fledged students. So because they are our workforce, we want to do for them.
The problem here is students have so much class in undergrad and postgrad. They're full of class, Cynthia. I think many of your classmates are having classes and classes.
Somehow we had to excite them to attend this, and like bribe them or something, like because this is good for them. It's good for that. Good for students means good for country, right?
I bribe them, and Luis here, Luis team, always there everywhere, and they like I like this kind of teams. I try to motivate them all the time, like let's go, and this is the only way to make them keeping into the flow, so that they don't feel one day I'm feeling Esteban is not here. I should also tell this. Adolfo is there from Esteban team. When I this idea came from Esteban. He is not today here, but in one day I just contacted Esteban in ResearchGate, and he was so kind. We need this kind of people more, and we had a meeting, and Esteban told, let us postulate some project, and what about and I told, I know Gustavo, and like we three started having this idea, and from there it generated, and then I came to know Joao in a workshop of Luis. So like this is the way we meet people, and they're so kind and collaborative, and we make it grow bigger. So Esteban is not here, but I also want to give a big thanks to Esteban. And Joao, please.
It was very nice to meet you all and to see what you're doing here in Chile. I have already said that, and the government of Chile, it's putting a lot of money in scientific research. I can't see that, and we can't lose this opportunity. So I am open for this. We are not in Brazil. We are not passing through a very scientific good thing. Our last government, it was terrible because he was saying that science is not important for humanity, and it's completely the opposite. But another thing that I would like to highlight is that I work with plant polysaccharides, but I am also trying to tackle several SDG from WHO, the Sustainable Development Goals. And one of the parts is the use of microorganisms to modify my polysaccharides. So that because they produce lots of hydrolases. And just a little chat, because I know that my postdoc, she's watching. I don't know if it's broadcasting yet, but she was watching, and she had isolated bacillus from the soil, and then we are now producing these bacillus, and it produces a lot of hydrolases, and then we are extracting the proteins in a very green way and using to modify my byproducts, so my food byproducts. So it everything related with circular economy. It's and I think it's one thing that here that in Brazil we need that. I don't know how it is the market here in Chile and how it is the byproducts maybe in the food market. I don't know.
It's growing.
It's growing, yeah.
It's growing in Chile, it's growing the interest for circularity, but also for food products.
Yeah.
So they're having an increase in the number of products and startups that are trying to set up new functional foods, new nutraceuticals, products, and things like that.
Whatever you want, and if you would like to write some prose about this, we can make it. And it was a funny thing because when we were coming with Fran from the airport, we stopped to eat, and in the dessert it was a jelly that I was looking at the ingredients, and it was not gelatin. It was a sulfated polysaccharide that was there, so it was vegan. And it's nice because in Brazil we don't have it. If it is a jelly, it's from gelatin, not from algae polysaccharide. So.
Food industry of Chile is the highest growing functional food industry in South America.
And you know what? It's just because I teach the labeling of foods. And I know that Chile, you are really ahead with the front labeling.
Even when in Chile, a few years ago, I have a meeting with some authorities from the health committee and so on, and they are not so open to try to to move forward on nutraceutical levels. For instance, in order to, and there are quite strict levels and norms following the states and the European community regarding nutraceutical claiming. So if you say this is nutraceutical or this is a functional food, you have to prove this is a functional food. It's the same like biostimulant. So you can say this is biostimulant. You have to prove that it is a real biostimulant in agriculture. But it is good regulations. Also, just help to filter good products. So.
Yeah, I know that the government in Chile, it's just like the Anvisa in Brazil. They are really restricted, but I think it has to be, because otherwise we are going to have, this is a protein bar that you're going, if you eat every day, you're going to be 100 years and no health problems. And it's not true. And it's different from United States, because United States FDA, it's just like, okay, you can put in the market, let's see what is going to happen. So, this modified citrus pectin, they had a very a big label that, it has anti-cancer effect. But there is no complex clinical trial with humans, and then FDA said, no, you need to take out. And then FDA took out. But in Brazil and in Chile, you can't put this in the market. So in FDA, I say, okay, you can put, let's see what is going to happen. And then, it's an anti-cancer. No, it's not anti-cancer. No. It's a dietary supplement, and it's fine.
And that's all.
And that's all.
And that's right.
Yeah, that's right. That's right. Yeah. Okay. Thank you, guys.
Thank you so much, everyone. Okay, to end up, I must say that this was like a dream come true for me. This proposal I always wanted to apply. So each one of you, I'm really thankful to. I will be emotional to talk about. And one thing I must tell, behind a lot of work that we have done in this project and a lot of students, I must thank Dr. Alex Echeverria and his confocal microscopy facility. Like he's one of the first year that in UCM he won first time a Fondeki project that is a huge project, and he was kind enough to incorporate me, and we started together working in UCM. So we share a lot of projects, INACH regular, Fondesit regular, and we do a lot of confocal microscopy of the microbial biofilms in his laboratory. So maybe after this we can go and see his confocal microscopy facility. And thank to all of you. If we can all take a photo, please, because we are officially ending the project, but we will be repeating this definitely.
You made it possible.
Yes, for sure.
Thank you for it.
Thank you.
Let's take a picture.
Yes.
I think it's better to
Maybe sit down. ¿Cómo está, compadre? ¿Todo bien? ¿Cómo está, socio? ¿Todo bien? Bien, bien. Gracias a ustedes para todo el streaming. Muchas gracias.
Gracias.